Lipocalins are small, low molecular weight proteins that exist in all three kingdoms of life, and there are 12 of them in humans. And Anicalins are nothing more than recombinantly engineered versions of lipocalins. Of these 12 lipocalins, they have a very, very low sequence identity, meaning that they share only a 20 to 25% sequence identity across all 12 lipocalins, yet they all fold into the same supersecondary structure, which for the sake of simplicity looks like a coffee cup. And that tells us that despite having a very, very low sequence identity, they're able to fold into the same structure, and they're very, very stable, and then they're excellent candidates for diversity, to make a diverse range of drug candidates. And so what we're able to do quite simply is to re-engineer the parts of the lipocalin that are involved in antigen binding, ligand binding, and create a re-engineered specificity against a host of targets, and yet expect them to still maintain their fold, to be a stable drug candidate, which is very important for functional screening and for safety purposes where you want to avoid things like immunogenicity. Given the broad applicability of the class and the robustness of the molecule, we are able to play in many, many areas. So, for example, we can go across the targeted biologic space that's common to, say, antibodies. We can operate in oncology, autoimmune, but we can also go into areas where, because of the robustness and the unique features of the molecule class, we can do things that other bulkier proteins like antibodies cannot do. So, for example, because these lipocalins naturally bind small molecule targets, we can engineer our molecules to go after very small, say, drugs themselves. So if you think about antidote applications where you want to guard against overdoses, or if you think about mopping up these ubiquitous proteins that are floating throughout the body, to antagonize those and get rid of them to treat such some things like metabolic disorders, we can go after completely new indications because we can bind small targets in the body. I think for Anicalin, the sky is really the limit. In 2010 and already at the beginning of 2011, we've made substantial progress both on our proprietary pipeline and establishing key therapeutic partnerships. So looking at our proprietary pipeline, that's very important to develop that to show the safety and the tolerability of Anicalins. As this is a cutting-edge therapeutic drug class, some companies would naturally have questions about things like safety and half-life, and through our phase 1 trial, we're able to have meaningful clinical data that's being generated as we speak that shows that this is a drug class that's not just a research tool, it actually has bona fide drug candidate properties such as safety, it's not immunogenic, and it actually stays in the body long enough to be a drug, and that's a significant advance on the technology to get pharma interested. The company was founded here based on the location of Professor Onescara, who has a laboratory department here at the Technical University around the corner. And that close proximity to Professor Scara has been an important part of the evolution of Pieris. Professor Scara is still an active board member, a collaborator, an advisor, and in addition to that, we have a broad talent pool to pull from in the area where we can always fill the ranks with talented people. Well, BioM has played a crucial role in the development of Pieris over the years. So first and foremost, they are an investor and a shareholder, and as such, through their input, we have been able to have facilitated business discussions and made some substantial progress with partnerships over the years. So looking at Professor Horst Damde and his contributions as an observer to our supervisory board, he has helped to put us in contact with a host of different groups of pharmaceutical companies, some of which have led to the culmination of deals that we've recently executed.